AIDS-defining illnesses, their causes and treatment. Last update, July 2001. Treatment recommendations based on the works of Dr. Heinrich Kremer, Hamburg, Prof. Alfred Hassig, Bern, Dr. Stefan Lanka, Stuttgart, Dr. Eleni Papadopoulos Eliopoulos et al., Perth, Dr. Leonard Herzenberg et al., Stanford University, and Furchgit and Ignaro, available at www.npi.nlm.nih.gov. The many and varied diseases that can define the AIDS syndrome. Fungal infections of the lung, of the mucous membranes, the brain, and the gut, and the degenerative changes in the endothelial cells of blood vessels and lymphatic vessels, Kaposi sarcoma, occur because of an ongoing heightened production of gaseous nitric oxide and oxygen radicals in immune cells and other cells. Under these conditions, CD4 helper cells mature predominantly to cells with Th2 cytic and profile, which migrate to the bone marrow where they activate defenses against bacteria by producing antibodies, but only few mature to Th1 cells measurable in plasma, which activate the detection and destruction of fungus and virus-infected cells, and of altered cells. If this situation persists, a higher quantity of proteins of the cytoskeleton and of mitochondria is released as an effect of heightened cell decay. Against these proteins a higher rate of the antibodies are formed. This antibodies and antibodies that occur in hepatitis and due to toxic pollution are detected by the HIV antibody tests. Once a certain, arbitrary level is reached, the patient is declared HIV positive. The elevated level of nitric oxide and oxygen radicals comes about as a result of ongoing contact with antigens, e.g. from repeated or chronic infections, injuries, operations and dirty water. Repeated contact of foreign proteins to the plasma, from coagulation proteins in blood preparations, and from semen liquid and unprotected anal intercourse. Contact to toxic substances in food, e.g. aflatoxins in wet cereals, medicaments and from environment pollution, toxic decomposition products from modern chemicals, heavy metals, e.g. carrier substances in hepatitis B vaccines, amalgam fillings. Inhalation of nitrites, poppers, which are stored in cells as NO2. They are released through physical exertion on increased exposure to calcium ions. This affects the endothelial cells of blood vessels and lymphatic vessels with a small capillary diameter, and leads thereby to degenerative changes, swollen lymph nodes and finally to Kaposi sarcoma. Impairment of the mitochondria, the single-cell energy suppliers, which synthesize the energy-carrying molecule ATP, used for all functions of the organism. The causes of chronic mitochondrial damage are damage of the mitochondrial DNA due to antibiotics, e.g. sulfa compounds such as cotrimoxazole, TMPSMX, which block the synthesization of folic acid and purine, and lead thereby to the exhaustion of the mitochondrial thyle pool. Likely effects are caused by heavy metals, and by cytostatics like CAST. All the substances bind the SH groups of glutathione and cysteine and impair thereby the activity of mitochondria. Reduced glutathione produced resulting from liver damage, e.g. chronic hepatitis, occurring frequently in gay men, hemophiliacs and intravenous drug consumers, excessive alcohol consumption, or through shortage of nutritional cysteine, ESP, in developing countries. Glutathione molecules reduce oxygen and nitric oxide molecules, so that ATP production in mitochondria is not disturbed. An ongoing shortage of glutathione means that phagocytes poison themselves attacking fungi and virus-containing cells by means of no reduced oxygen transport in cells because of oxidation methemoglobinemia, which exceeds the reductive capacity of glutathione. This comes about because of the strongly oxidizing effect of nitrites, poppers, antibiotics, cotrimoxazole TMPSMX, and insecticides, e.g. linden in moistures against crab louse, nucleoside analogs, e.g. AST, heavy metals and chemicals. Lack of plant antioxidants which bind to toxic degradation products, oxygen radicals, and thereby reduce inflammation and stress reactions. On prolonged impairment of mitochondria, they dissolve their symbiosis with the host, Warburg phenomenon. Cells then increasingly switch over to producing energy by anaerobic fermentation, which results in excess lactic acid production, and the growth of fungi and opportunists, and ultimately to wasting, at which point cells obtain essential nutrients directly from the myoprotein. By an heightened activity of reverse transcription, the cell nucleus then saves its genotype. Continuous activation of macrophages leads in this situation to an ongoing release of messenger substances, interleukin-2, which trigger the release of stress hormones in the adrenal gland. These hormones induce the formation of Th2CD4 cells that activate the formation of antibodies in the bone marrow, whereas cellular immune reactions induced by Th1 cells are continuously suppressed. By means of a supply of sulfur compounds in sea salt, mineral water and algal products, and of cysteine and methionine containing protein mixtures, cysteine, N-acetylcysteine and arginine, 3 to 8 gram daily, right parenthesis also incurred and wet.